welcome to my YouTube channel. My name is Saj Gowde. Today we will be exploring the world of biomolecules. Our main focus will be on proteins, their structure, functions, and analytical techniques. First, let's move on to the basics, that is biomolecules. Biomolecules are chemical organic compounds found in living organisms that are essential for life. They are made up of elements like carbon, hydrogen, oxygen, nitrogen, and phosphorus. There are majorly four types of biomolecules that are lipids, carbohydrates, proteins, and nucleic acids. Proteins are polypeptides. They are also known as heteropolymers because they are made up of two or more monomers repeating n number of times. Each protein is a polymer of amino acids. Amino acids are linked by a peptide bond which is formed when the carboxyl group of one amino acid reacts with other amino group of another amino acid. As there are 20 types of amino acids, several other amino acids have side chains with positive or negative charges, while others have polar or uncharged side chains. They differ from each other in R groups, which vary in structure, size, and electric charge, which influence the solubility of these amino acids in water. These side chain chemistry also influences the chemical properties of a protein. Charged amino acid side chains can form ionic bonds, while polar amino acids are capable of forming hydrogen bonds, which make the protein soluble in water, whereas generally proteins are not very freely soluble. Let's first talk about the structure of proteins. There are four levels of structure. Firstly, the primary structure. It is a specific linear sequence of amino acids in a polypeptide chain. This primary structure of a protein helps in determining the gene code which is read from a corresponding gene. This is the most unstable structure of protein. So even though the polypeptide chains are linear, these further fold and coil into three-dimensional shapes, giving proteins a unique and stable spatial structures which will be seen further. Even a small change in one of the amino acids can lead to mutations. For example, sickle cell anemia, where a single amino acid change can affect the function of hemoglobin. As further we see the secondary structure, which is a local folding into alpha helix and beta pleated sheets. Alpha helix are right-handed coiled conformations by peptide bonds. These structures stabilized by hydrogen bonds formed between the carbonyl oxygen from one amino acid and amide hydrogen of another, typically 4 residues away in the sequence. Most of the amino acids that we see are methionine, alanine, glutamine, etc. Beta pleated sheets are formed by the alignment of polypeptide chains that are formed together, held together by hydrogen bonds between polypeptide backbones which resemble a pleated sheet. These strands can run either parallel or anti-parallel to each other. As further we see, these secondary structures then form a further complex bending of single polypeptide chain, which is stabilized by interactions between different types of bonds like hydrogen bonds, disulfide bridges, hydrophobic interactions, etc. It gives an overall 3D arrangement to a protein structure. Further, we see that quaternary structure, which is an assembly of multiple polypeptide subunits held together. It is the most stable structure of protein. Next, we see the functions of these proteins with respect to their composition and how they play a critical role in most of the biological processes. Firstly, enzymes. Enzymes help in catalyzation of biochemical and metabolic reactions, for example, DNA polymerase, amylase. Then we see our structural proteins, which provide support and shape to cells and tissues, example, collagen, keratin. Then we have the regulation proteins, which regulate gene expressions and translations processes, such as transcriptase factors, hormones like insulin. Then we have defense proteins that protect the body from pathogens, example, antibodies. Contractile, which means movement, they help in movement of these proteins. Storage proteins help in storing amino acids or essential ions. Transport proteins are proteins that carry molecules from one membrane to another. For example, the integral proteins in a lipid bilayer. Signal transduction is a function where it receives and transmits these signal messages within or between the cells. For example, hormone receptors, G protein coupled receptors. As we further see, we have several methods which help in the isolation and analyzing of proteins based on their physical and chemical properties. The most commonly used method is gel electrophoresis. The term electrophoresis describes the migration of a charged particle under the influence of an electric field. Electrophoresis can be done in two ways, vertical and horizontal. PAGE, which is the abbreviation for polyacrylamide gel electrophoresis, is mostly used for separation of proteins. Polyacrylamide gel is thermostable, transparent, durable, and a chemically inert compound. There are two types of PAGE electrophoresis used for separation of proteins. There's STS PAGE and native PAGE. Today we will be focusing more on the STS PAGE theory. STS PAGE is the most widely used method for analyzing protein mixtures quantitatively. The apparatus needed to perform STS PAGE on protein samples are electrophoretic support for separation, buffer, stacking gel, separating gel, electrodes, cathode and anode, and a power supply. STS PAGE separates proteins based on size or molecular weight and is used for qualitative analysis, purity checking, and molecular mass determination. The roles of STS, which is sodium dodecyl sulfate, is that they bind to proteins and denature them, which gives them a uniform negative charge since it is an anionic detergent. It is also used with pair with beta-mercaptoethanol, which helps reduce the disulfide bonds between proteins and further denatures the proteins into units. The proteins become rod-shaped, uniformly negatively charged, and separated solely based on size, not shape, not charge. The stacking gel, which is used, helps in concentrating the protein bands into sharp zones and also helps them to align in a thin, sharp band before fast moving chlorine ions and show slow glycinate ions. Separating gels is helping in separating the proteins by size as they migrate towards the anode. Smaller proteins move faster through the molecular sieve of the gel electrics. For visualization, we use Kumasi blue to stain these samples, which reveal bands on the gel. They can be compared to the standard samples and helps in determining the molecular weight of proteins. Even molecules with similar charges will begin to separate if they have different molecular sizes, and they will experience different frictional forces. And another technique that is most commonly used is chromatography. Chromatography is a laboratory technique which helps in separating proteins. It has two phases, a stationary phase and a mobile phase. Three main types of chromatography that is mainly used for separation of proteins is size exclusion chromatography, ion exchange chromatography, and affinity chromatography. In size exclusion chromatography, it is mostly separated by molecular size of the protein. Ion exchange chromatography is done by separating these proteins due to their charges. Affinity chromatography separates proteins based on a specific and reversible interaction between a target protein and a ligand attached to the stationary phase. For example, antibody, antigen, enzyme substrate, etc. On to why these techniques are suitable. High specificity of these techniques help in isolating these proteins with a minimal contamination. Then scalability and reproducibility, which means it helps and is useful in both research and industrial settings. Then we have the quantitative and qualitative analysis. STS PAGE and spectrophotometry provide both present and quantity information that can be used for further research purposes. Then the other point is preserving protein function. In some cases, non-denaturing methods can retain the protein activity for further functional studies. Last but not the least, versatile. These different techniques can be used combined, and for example, the 2D electrophoresis and chromatography together for comprehensive analysis. Understanding protein structure and function is crucial for the relevance of proteins in today's world. As we can see, disease research has helped in making drugs that are protein or protein-based, targeting proteins with specific inhibitors or activators. By studying protein, it also helps in understanding the cellular pathways and environment. In biotechnology, enzyme production is of utmost importance, which helps in using it for diagnostics, therapies, and industries. Cell biology also helps in investigating how cells grow, divide, and respond to their environment. By understanding this, we can say that proteins are involved in every cellular processes, making them essential for life. Thank you.